broadcast is now starting. All attendees are in listen-only mode.
Hello, everyone, and welcome to Fruit and Vegetable Magazine's webinar event on maximizing biological fungicides. My name is Jennifer Page, and I'm the editor of Fruit and Vegetable Magazine, and I would like to thank everyone for joining us today. In today's webinar, our presenter, Anne McCrae, will be discussing how biological fungicides work and how you can maximize their performance and consistency on your own farm. Anne is a technical specialist with BASF and is a key contact for Canadian fruit and vegetable extension personnel. She also provides technical support to BASF representatives across the country. Welcome, Anne. Before we begin, there's just a few housekeeping items. So we will hold a question and answer period at the end of the presentation. And we encourage you in the meantime during the presentation to type your questions in the text box in the GoToWebinar panel. We will be collecting those questions throughout the presentation, and then we will ask them once the presentation has concluded. This session is being recorded and will be sent to those who have registered in a follow-up email 24 hours after
formulation first to receive and send the correct signals. Hundreds of metabolites can be produced by the spores and are based on your environmental factors. Two of the key metabolites produced are surfactant and niturin, and I will discuss more about those in greater detail sort of later in this presentation. So when we look at what inhibits, so what happens when this communication is obstructed, you have what's called miscues. The spores can no longer work as one community. And when do these miscues happen? When we try to force these living organisms to function contrary to their nature. So we cannot control nature. That's the first thing we all need to understand about biologicals. The best we can do is know as much as we can about how the organisms work and provide them with the best parameters to ensure a successful outcome. We can influence nature, but we certainly cannot guarantee it. So I've made up these sort of very simple diagrams to help us understand a little better how these miscues in communications happen when we apply these bacterial spores ourselves. The black dots represent spores and the triangles represent metabolites, so fairly straightforward. When a biological fungicide is packaged, metabolites cannot be guaranteed to the type and concentration. They could be the wrong metabolite at the wrong time, and even if it were the correct metabolite, a low population density may not receive the signal. In the first diagram, this is a case of low spore population density. The spores produce metabolites to communicate, but no one is there to receive the signal. In the middle picture, the spores are in good proximity to each other. However, there are too many conflicting signals, and the colony cannot act as a whole to either grow or protect itself. Therefore, your formulation of your biological fungicide really matters. The closer you can get to a product that has pure live spores and not a lot of extra junk such as dead cells and extra metabolites, the better chance it will have to be like this third picture on the right side, where there are a high number of spores, which then produce the same metabolite, all communicating the same thing, whether that is to grow or feed itself or to protect against pathogens. So with the knowledge BASF has collected, we have tried to set a quality standard for this purity of spores that will help us to sort of enhance the performance and reliability. And so with that, I introduced Serifel, which is our new foliar applied bacillus fungicide. It's called Bacillus amlia liquefactions, and it is a special strain called MBI 600. We've done extensive testing on Serifel on how it works and some of the metabolites it produces and how it communicates. For Serifel, on its current label, it is only registered on grapes, and it's registered for the suppression of powdery mildew and botrytis. As I said, we've done a lot of research to work with Serifel to help us better understand this, and sort of with this presentation, I wanted to go through some of that research we've looked at, which will help us better understand Serifel and better understand how biologicals work and how we can work with them. So with what we have learned already about biologicals and their communication, we know that when you have the right metabolites working as nature intended, you have predictive behaviors. So you know how this will work as soon as it is applied to the leaf. This diagram shows those predictive behaviors of Serifel. So on the top right-hand side, we have Serifel is applied to the plant. After that, usually when the temperature is around the 10 to 20 degrees Celsius, Serifel spores will produce a metabolite called surfactant. This surfactant metabolite communicates to the spores that it needs to grow and be mobile. As temperature increases and as the colony increases at that 20 to 30 degrees Celsius, the Serifel spores will then produce a metabolite called iturin. And iturin is the key fungicidal, bactericidal metabolite. At a temperature of 30 to 50 degrees Celsius, your pathogens are generally dormant. However, Serifel will still continue to grow, albeit maybe slower, but will still be there protecting your plant. So what that kind of explains is Serifel's three key modes of action. So the first and second are fairly straightforward. Serifel, when it grows, creates a physical barrier. So Serifel spores reproduce and occupy space in the plant, so disease-causing pathogens don't have any room to be there. They don't have any room to grow. With that, Serifel spores will continue to grow, and then there are not enough nutrients to support the pathogen. So the Serifel spores take up all the nutrients. And thirdly, and probably most interesting, is once on the plant, the spores germinate and produce metabolites that have fungicidal and bactericidal activity. So they will actually attack the pathogens. First and second modes of action are fairly straightforward, and this just gives some neat pictures showing Serifel spores colonizing on the plant tissue. Basically, these robust spores grow and reproduce on the surface of the plant and create a complete physical barrier. And with this, they're excluding all of the pathogens and also using up all of the nutrients. So there's limited nutrients for other pathogens to grow. Thirdly, and most interesting, is the third mode of action, which involves breaking down the pathogen cell. A healthy pathogen cell comes in contact with Serifel metabolites. The metabolites insert themselves into the pathogen cell membrane, and this causes the pathogen cell membrane to collapse. We have kind of a neat picture to show that. This is a picture using an electron microscope of a powdery mildew pathogen, and the Serifel metabolites that make holes in the membrane of the mycelium. With the damaged membrane, or the membrane that has broken down, the powdery mildew pathogen can no longer function properly. So kind of a neat way for Serifel to protect itself. The next thing I wanted to talk about is reading a bacillus-based fungicide label. Unlike traditional fungicide labels where you look at the amount of active ingredient, usually in grams per liter or as a percentage, a bacillus-based product will give you CFUs per gram. CFU stands for colony forming units. This is the number of live spores in each gram of product. Live spores that are available to colonize the leaf, compete with pathogens for nutrients, and create fungicidal metabolites. With Serifel, we wanted to pack in as many live spores as possible. There are actually 55 billion CFUs, or live spores, in 1 gram of Serifel. So if we think back to the diagram with the circles and the triangles, we remember pure formulation matters because we need the right metabolite produced at the right time and more metabolites produced at any given time. Therefore, the CFUs, or the live spores, means less chance of miscues or miscommunication within the spore community as a whole. With all the bacterial spores communicating as one, we get predictive behaviors, and it gives a bit of a head start. So it's able to act more like nature. Here is how we see that head start sort of working. The Serifel spores adapt more quickly to the environment and produce metabolites suited to the ambient conditions in the petri dish. As you can see in the picture, Serifel, that's on the right-hand side, demonstrates greater leaf surface colonization at only 10 degrees Celsius. Bacteria typically prefer much warmer temperatures, but even at this cool temperature, Serifel can communicate as a colony and begin to grow. As you see in the little bit there, if you look at the petri dish, it's in the sort of bottom left-hand corner. It's all starting to grow. Along with the head start advantage, pure spore formulation and good communication make Serifel more adaptable to environmental conditions. Serifel has a rain-fast of 3 hours. It was tested under intense UV light at the equator and able to tolerate it for 7 days. And Serifel can also withstand temperatures exceeding 50 degrees Celsius. If we take a closer look at effects on temperature, this graph shows the growth of Serifel as the temperature increases from 15 degrees Celsius to 40 degrees Celsius. The optimal temperature for bacteria multiplication is between 25 and 35 degrees, as you can see here, where we should see the most growth, which is also optimal temperatures for most pathogens to grow. Therefore, your biological fungicide works best when conditions are conducive to their growth and the pathogen's growth. So they grow more as those pathogens are growing more. As mentioned before, one of the metabolites produced by Serifel is called surfactant. The name for this metabolite is not a coincidence. It was named this because it acts like a surfactant, and it helps the fungicide to stick to the plant. The more surfactant metabolite produced, the better it will stay on the leaf. And as we see in the graph, the dark green bars are Serifel. So at that lower, it likes to produce Serifel at those lower temperatures. So you see those really high numbers at lower temperatures, but then it continues to produce surfactant at the warmer temperatures as well. Surfactant can also be an antifungal metabolite, but its key communication is for motility and growth, as well as acting as a rain-fast enhancer. Serifel has a rain-fast of 3 hours. As you can see in the graph, after a rainfall event of 20 millimeters, the competitor washed off the plant, but Serifel remained and was still able to control botrytis. So that's a key thing with one of its metabolites, surfactant, is that it's helping it stick to the plant. So we do have a rain fast of 3 hours. Here's an interesting lab experiment done to show how Serifel spores actually create an exclusion zone. So a fungal lawn of botrytis was grown on the underside of a petri dish lid. The dish was then closed, and spores were allowed to fall down onto the media where the Serifel spores were growing. Spores that fell within the ring of produced metabolites were not able to germinate. So even outside of where you can see the actual Serifel spores, you see there's no botrytis growing because of these metabolites that are creating this exclusion zone around it, protecting it. So this was another lab test done to show the importance of applying Serifel preventatively. So as I mentioned before, we have these Serifel spores. In the first petri dish, this is what a curative application would look like. So if a pathogen was placed on the petri dish first and allowed to establish, and then Serifel was applied after, You can see the disease had lots of time to take over, and Serifel wasn't able to compete with the disease. When you look at the middle Petri dish, the application was at infection. So the disease was placed on the Petri dish at the same time that the Serifel spores were. And as you can see, the disease was still able to grow, but Serifel had time to colonize and create that zone of exclusion and protect itself and fight off, but you're still seeing some of the disease. On the right-hand side in the Petri dish, it was a preventative application. So Serifel was put there to colonize before the disease was. And as you see, it's our best case scenario where Serifel was really given a chance to fight off this disease. So it's very important when applying your biological fungicide that we do it preventatively before there's any sign of disease. Previously, we have spoken about surfactant, a key metabolite produced at lower temperatures. A second metabolite is iturin, which is the key fungicidal metabolite. Iturin is produced generally in warmer temperatures, which are conducive to pathogen growth. This graph shows that Serifel has the most iturin produced at 25 degrees Celsius, but unlike its competitors, it also gets a head start and produces this key fungicidal metabolite at 15 degrees Celsius. Next, I have some in-field trial data that I wanted to show you that we've collected. This graph shows the summary of 6 BASF trials rating the efficacy of Serifel at high and low rates. On the Serifel label, there will be a rate range from 250 grams per hectare to 500 grams per hectare, and we recommend using the high rate at a 7-day interval for more consistent, better control of powdery mildew. I also had, this past summer, I was able to see some of our trials firsthand that were done from some third-party research companies. In this vineyard, we had some very high powdery mildew pressure, and the Serifel program that we had was doing an excellent job of controlling that powdery mildew. Serifel also has activity on botrytis. Although in our trials, we have found it is not as consistent or as strong as it is with powdery mildew. Botrytis can be a very nasty disease to control. And so for botrytis, Serifel can be used alone for suppression, but it can also be used with greater success if it's used as a tank mix. And I'm going to talk a little bit more about that. One exciting thing we have discovered about Serifel is its tank mix compatibility with other traditional fungicides. Serifel can be tank mixed with conventional chemistries to enhance performance and manage resistance. This can help to prolong the longevity of conventional chemistries by helping to delay the development of resistance. And it is also proven to manage some diseases already resistant to chemical fungicides. So if we take a closer look at what this means and what happens when Serifel is tank mixed with certain fungicides, we see that some of these fungicides can apply a slight stress to Serifel without hindering growth. Also, the stress triggers additional growth for improved coverage and stability and the production of antifungal metabolites. So in these pictures, it kind of shows what's happening. On the left-hand side, the picture shows Serifel on its own. The sort of dark red lines are the spores, and then the light pinkier lines are all the metabolites. When Serifel was tank mixed with Canthus, which is in the center picture, we see a lot more spores and a lot more metabolites, which is a pretty exciting reaction. Considering we are mixing a bacteria, these spores with the fungicide. So it seems to give it this boost and makes it work even harder to protect our plants. And the same happened when Serifel was mixed with pristine, which is in the right-hand picture. We also looked at some field studies to see how this would work when we tank mix Serifel, and to see if it would complement some chemical crop protection programs. So in the first two bars, we have a competitor and Serifel on their own, and this was when they were applied at a 7-day interval. The other 4 bars were done at a 14-day interval. The first product used was enhexamide, which is actually not one that is typically used for powdery mildew because it doesn't perform very well. But when we tank mixed it with Serifel, we got over 90% control. It made it look like both products were getting this boost. As well, we tank mixed Serifel with Vivando, and Vivando typically is a really strong product on its own. In this trial, we were looking at 97 or 98% control, but when we add Serifel, it still gave it an extra added bonus. So you got 100% control of your powdery mildew, which is pretty exciting. So here is a list of all tank mixes we have confirmed to be compatible with Serifel. As in, they will not kill the spores when you mix them together in a spray tank. However, not all of these will necessarily give Serifel the boost you saw in the previous slide. That boost seemed to occur most often with group 7 chemistries, with products such as Cantus and Pristine. But all of these products, there was no issues with tank mixing. Serifel still worked really well, and there was no mixing issues when spraying either. So there's lots of products on here. They're all fungicides, insecticides. We have some copper, sulfurs, any surfactants, adjuvants, no issues tank mixing. Which brings us to the next slide. Although we have an extensive list of products that are compatible with Serifel, there are a few key fungicides that are not, like the 4 listed here. So your chlorothalonil, like Bravo, mancozeb, metalaxyl, like Ridamil, and your Metaram or Polyram. These should not be tank mixed with Serifel, as they will kill the living spores. If you have any of these products in your rotation, you should allow for 7 to 10 days between your Serifel applications. So these are the 4 important ones. So now that we know what we can and cannot tank mix with Serifel, let's talk a little bit about resistance management. Serifel is classified as a frac group 44. So it can be rotated with other frac groups in accordance with your IPM recommendations. Because this is a unique group and a biological, we get to reduce selection pressure. So replacing a conventional fungicide with Serifel, and the spray program can reduce the pressure on that fungicide's mode of action without compromising any efficacy. And then obviously helping control resistant pathogens. So field trial data demonstrates Serifel provides equivalent control of susceptible strains of botrytis. And we have taken a look at that in some of our trials as well. In this case, we took a look at a group 7 sensitive strain and a group 7 tolerant strain. And obviously, when it was applied with just a group 7, we did not get the control on the group 7 tolerant strain, but we still had the same control when using Serifel. So the next section, and sort of lastly, I'm going to cover some best management practices and also sort of how to use tank mix and ease of use for Serifel as well. And the first thing, which sometimes we don't often think about with a biological, but is very important, is that you should still use personal protective equipment. So all microbial or biological pesticides do have the potential to be skin sensitizers. So we should make sure we are wearing our personal protective equipment like gloves, long sleeves, pants, boots, apron, if you have it. So even though this isn't a traditional chemistry, it's important to remember that. So ease of use with Serifel. So this is another exciting thing with this new product, is that we found we're getting less clumping and less foaming. There's a lower use rate, which makes Serifel just an easier product to handle. And also with that stays in suspension longer. So you get fewer tank cleanouts and mixing issues with Serifel. So in these photos, Serifel was mixed with water and allowed to settle overnight. And it's just showing how it tends to stay in suspension much longer than other competitors. With tank mixing, I'm going to go over this a little bit because there's something that's a little different than when you're mixing some traditional chemistries. Obviously, the beginning is fairly straightforward. Always make sure your spray tank is clean. Next, we partially fill the tank with water and start your agitation. So pretty similar to what you would normally do. The difference is that before you dump your Serifel into the tank, we recommend creating a slurry. So we recommend taking a bucket with water and dumping the Serifel in that bucket of water and mixing it up. And we call this a slurry. So then you would dump that slurry into your spray tank that's currently is agitating. So you add your Serifel slurry, and then you would add whatever tank mix partner that you're using or adjuvant if you would like to, and then you continue that agitation, obviously, and you can go out and spray. We don't recommend leaving Serifel in the tank for long periods of time or overnight. And then obviously when you're done, make sure you properly clean everything out. All of these instructions will be on the label, so you're definitely welcome to check out the Serifel label just if you're not 100% sure. So a few of the management considerations for Serifel. As I said before, we will have a rate range, but we recommend that 0.5 kilograms per hectare just for better consistency when using it. Ensuring thorough coverage is very important, so make sure those water volumes are high enough that you're going to cover that whole plant, because it's important to get as many of those live spores covering all of the plants, because that's part of the way that they can control those pathogens, is that they're able to spread and cover everything. There shouldn't be any issues with your pH of your water. We are okay with a pH between 4 and 9. And the use of a surfactant can improve performance just because that generally improves the coverage on your plant. So it may not be necessary, but it certainly won't hurt it. And if you're worried about that coverage, it doesn't hurt to have surfactant. So as I said before, rain fast is 3 hours. Your re-entry interval is only 4 hours with the biological, and the pre-harvest interval is 0 days, of course. And also the added bonus is it's exempt from MRL status because there's no residues. In case anyone was interested, I wanted to just show everybody kind of what the packaging looks like. They come in 4 small containers, which go inside one box, pretty easy to use and open up, which is kind of neat. And if you're using that high rate, each one of those little containers will treat about 10 acres. So now I just want to review what I've talked about with Serifel, just go over some of the key points. First of all, Serifel is registered on grapes only and for the suppression of botrytis and powdery mildew. The really neat thing about this is it complements chemistry-based programs. So this is great as a tank mix partner and helps manage potential for resistance. It does not add any recordable residue. Serifel allows for greater flexibility by extending the window of application right up to the end of the year, right up to harvest. And we are trying to set this quality standard for concentration and purity, which, as I at least able to stay awake through all of this interesting biological talk. If you're looking for any additional information on Serifel, or you want to check out that label, just go to our website at www.eggsolutions.ca/horticulture. And that is all for me, and I will turn it back to Jennifer. Thanks.
Great, thank you so much for the presentation, Anne. We'll now proceed into the Q&A session. So the question we have here, is there any interest in testing Serifel on apple trees for powdery mildew?
Yeah, good question. So currently our label will only be for grapes, but BASF is continuing to look at other fruit and vegetable crops and other uses for this product. So yes, we are still in the process of doing that research, but it's certainly on our list of things to look at.
All right. The next question, are there buffer zone restrictions when using Serifel?
There are not, which is the nice thing about Serifel, because this is a biological product and it's found in nature, then there's no issues with spraying. We don't have any issues with buffer zones to any waterways or forested areas or anything like that.
Can this be used on high bush blueberries for mummyberry?
So because our label will only have grapes on it, then it will not be able to be used for blueberries yet. But it doesn't mean that, as I said before, we will continue to work with this product and hopefully have label expansions in the future. So it's certainly one that we can take a look at.
What is the relationship between Serifel to Hinga's effective microbes, which is a whole mixture of multiple organisms?
So what is the relationship? Or?
Yeah, the relationship between Serifel and the effective microbes.
I guess, do you mean as in like other microbes that it would come in contact with, when we apply it? Or microbes like different pathogens that it might be attacking?
I believe when you apply it.
Okay, I would say, well, I guess we can't say 100% sure because we haven't looked at everything that it interacts with. There are enough number of different pathogens other than powdery mildew and botrytis that are being looked at that looks very promising. It will also attack. As far as beneficial ones, I am not sure. It's not something I have looked into yet, but it's certainly something I can look into and get back to you.
Is this safe for human consumption of grape leaves in restaurant use?
Human consumption of grape leaves?
Yeah, that's the question.
That's a good question. I don't know. So because it has, I mean, if you treat it like anything else, the pre-harvest interval is 0, and you have a re-entry of 4 hours, so you can't go in and touch anything for the 4 hours. But after those 4 hours, there's no issues with with harvesting or anything like that with touching the crop. So I'm not sure if that answers the question, but that's kind of what the what the label states.
Is this listed for organics with the Organic Materials Review Institute?
Yes, it is. Good question. So we do have the EcoCert certification for Canada for organic, and the OMRI certification in the US as well. So yes, it is.
And does Serifel have activity on any other diseases?
So we're doing extensive testing for other diseases. Currently, obviously, as I said, Serifel will be registered for powdery mildew and botrytis, but there are a number of diseases that it also controls in other crops, and so we're going to continue to look at the efficacy of that and try to expand this label as well. So stay tuned in future years to hear a little bit more about that.
Are all biological products made the same?
So they are not. Well, each biological, although it is a bacillus, if we're talking about bacillus-based biologicals, have their own unique formulation and strain. So they are, in our case, our strain is the MBI 600, which is unique, makes it unique, but also the way that the product's produced or packaged, as I talked, there's a lot of products that can have a lot of stuff in them, but not necessarily live spores. So you can get a lot of extra dead tissue, dead spores or metabolites and things like that that aren't useful when you're using products. So it's important to take a look at how many actual live spores you have in the product that you're using.
Does air humidity have any effect on longevity of Serifel once applied?
So the testing done to look at like high heat temperatures did show that it will maybe not grow as quickly when you have those high temperatures and low humidity, but it will not kill it. So it may go dormant, but it will still be present for when you have that moisture and those temperatures decrease.
Can you leave Serifel in the spray tank overnight?
So our recommendation is to not allow the spray mix to stand for prolonged periods. And so especially we recommend not to leave it in the tank overnight. So if you can, make sure as soon as you mix your Serifel, then try to spray it all out.
I think that's all the time we have for today. Once again, I'd like to thank Anne so much for her presentation. I'd also like to thank BASF for sponsoring this webinar presentation. As I mentioned earlier, this session has been recorded and will be sent to those who have registered in a follow-up email. The recording will also be available on our website at fruitandveggie.com. Fruit and Vegetable Magazine will be hosting another webinar presentation on understanding and managing aphids on March 11th at 2 p.m. Eastern. Please visit our website for more information and to register. I hope you all enjoyed the presentation today. Thank you so much for joining us.